And now we welcome Professor Julie Fitzpatrick from the Morden Research Institute, who is going to tell us a little bit about the multiple interactions that take place that start in the soil, and how that is then ingested by the animal, and how the interactive cycle relates back then ultimately to human health. Julie, thank you very much for coming along today. You're the Scientific Director of the Morden Research Institute and Chief Executive of the Morden Foundation. You're also Chair in Food Security in the College of Medical, Veterinary and Life Sciences at the University of Glasgow. Much of your own research is focused on livestock health and disease prevention, both in the UK and overseas in developing countries. Can you tell us in what way you think soils contribute to an animal's environment, and where is the biggest challenge, or at least some of the challenges, in providing this as a quality environment?
Well, soils are obviously very important, particularly for ruminant species which graze grass and convert that inedible substance in human terms into a product that can be used by humans, whether it is meat or milk. And so the quality of soils is very important for the quality of the grasses and also the crop products that ruminants go on to use. For example, in Scotland, 80% of our landmass is considered less favoured area, so it's mainly upland and hill land, which is ideal for grazing ruminants.
Thank you. What role do you think soil plays, though, in the persistence and transmission of disease in animals?
Soil is involved in the life cycles and in the transmission of a number of important animal diseases. A good example is toxoplasmosis, which is caused by the protozoan parasite Toxoplasma gondii. This is an important infection which has the cat as the definitive host. So the cat passes out the infective stage, the oocyst, in faeces, and can produce hundreds of millions of oocysts during active infection. That's then a risk for humans who come into contact with cat faeces directly. However, cat faeces can also contaminate things like vegetables and plants, animal feed and water, and in that way other species can also become involved. For example, persistence in soil can last as long as 2 years. So animals picking up infection from soils include particularly pigs that are kept in organic or extensive systems. So they become infected, and humans become infected from eating those meats. If they're undercooked, they become exposed to tissue cysts within the pig meat and can go on to develop problems.
There are so many ways that the soil interconnects with the animal and also back to the human. But can you provide an example direct from soil to humans and the problems with human disease potentially?
It's perhaps not direct, but certainly one of the most important pathogens is E. coli 0157, which is a common microorganism, bacterium, which is shed from cattle across the world, but particularly there have been some serious outbreaks in Scotland. The bacteria E. coli 0157 can contaminate meat at the point of slaughter and can be transferred to humans again through handling infected meat or through insufficient cooking of meat. But E. coli can also transfer via faeces and via manures into the soil substrate. And within soils it's known that E. coli can be taken up by plant systems and other vegetables and can again become a route of infection for humans.
And you know, in this whole seminar today we're talking about soil biodiversity, and there's been a whole range of different organisms we've been discussing. Have you got a favourite?
Well, I do have a favourite, but it's an animal pathogen that is a contaminant of soil, as opposed to one of the good guys, one of the good microorganisms, which I imagine other people might choose. The one that I'm particularly interested in is the cause of a disease called Johne's disease, which is a fatal diarrhoea of ruminant species. It's caused by Mycobacterium avium paratuberculosis, a very thick-walled bacterium, and we know that it persists in soils for considerable periods of time, up to a year. So that means even though we try to diagnose the disease and deal with it in the animals, we have this sort of hidden bacterium surviving in soils and making disease control very difficult. Recent findings have indicated that it's possible that Acanthamoeba, which is a free-living soil organism, can take up this MAP bacteria into its intracellular position, and there it can act as a sort of Trojan horse for Johne's disease. So the MAP bacteria are thought to replicate within the amoeba and also to upregulate some of their virulence factors. So when they're then released from these amoeba, that they're reintroduced to the soils and exposed then to the ruminants who graze on those soils. So a very good example of where disease control has been difficult because perhaps we haven't taken the interaction with soil sufficiently seriously.
Thank you very much, Julie, and good luck with all the research that you're doing and to help making a mark in the field of global food security. Thank you.